All right, everybody. Welcome back to the digestive system. This is the third part of this very long lecture. This will be the shortest part. Um, the way we arrived here to this part of the digestive system, if you look at this diagram on this slide, a little bit of the ileum is left here. Um, our last large segment, the third workshop of our digestive system, was the small intestine. Using pancreatic enzymes, using that emulsifier from bio, we have successfully degraded all proteins to amino acids, all carbohydrates to simple sugars. We degraded triglycerides to fatty acids and glycerol. We looked at the absorption process happening in the small intestine. And so here at the end of the ileum, there is a stream of fluid that has some water in it, electrolytes, all the undigested or unabsorbed material, and all that is streaming because of the peristalsis of the small intestine, streaming into the large intestine. So anatomically speaking, the large intestine has its own parts to it. If you remember, the small intestine had the duodenum, jejunum, and ileum parts to it. The large intestine has the cecum, this pouch-like area here. We have the colon, and even in the colon, within the colon, we have the ascending, transverse, and descending colon, the sigmoid colon, and finally the rectum, where the fecal material collects and ready for defecation. In terms of tissues making up the large intestine, we're not going to see anything tremendously different from the stomach through the small intestine and now through the large intestine. The mucosa is simple columnar epithelium. In the large intestine, there are copious mucus production to provide lubrication for the fecal material that is drying out as it's moving through the colon and eventually to the rectum. The muscularis has smooth muscles. We have seen that in the stomach, in the small intestine. There is one peculiar function of the muscularis that smooth muscles form thick bands, and you can see the thick band right here and also on the top here on the transverse colon. Looks like a little rope, and these bands of smooth muscles called Taeniae coli, they are going to move fecal material at surprising speed from the ascending through the transverse through the descending colon and move it into the rectum. So quick movement of fecal material will be seeing a reflex that is utilizing these muscles. So Taeniae coli are bands of smooth muscle. They are there for quick movements. There are also thinner layer of smooth muscles around these cheek-looking puffed portions of the large intestine. These individual puffs are looking like a cup from the inside. So each individual cup is called a haustra, and smooth muscle in the wall of the haustra creates a mixing motion, and the last bit of absorption is happening or facilitated by the movement of the haustra. The large intestine is a very, very uncomplicated part of our digestive system. So mechanical digestion, this is all about moving fecal material. We have peristalsis. We have the churning of the haustra. Individual cups are mixing material to enhance absorption. And then here is the use for the Taenia coli, the gastrocolic reflex. So using the enteric nervous system, this cross-talk conversation between different parts of our digestive system, stretch receptors in the stomach can start a reflex. Putting large amounts of drinks and food items into the stomach will make the stomach to feel compelled to inform the colon to empty the colon as much as possible, move fecal material into the rectum, make room for the incoming material. So filling the stomach will cause a movement, peristalsis, utilizing the Taenia coli, and large amount of fecal material will be moved from the colon into the rectum. So this mass peristalsis is an example of conversation between the stomach and the colon using the nervous system. And it also gives an example of peristalsis and the use of the Taenia coli in this part of the digestive system. When it comes to chemical digestion, we have a simple task here because we are not adding new human enzymes. Mucus secretion is very, very high, providing lubrication. Chemical digestion, degradation of organic nutrients is done by bacteria. And here's a list of what these bacteria do. If any carbohydrate was turned or was unabsorbable or undigestible, bacteria create gas. Undigested proteins can be turned into smelly compounds. Odor comes a lot of times from undigested proteins being degraded by bacteria. And here's a little more specific example. Bilirubin, and we looked at this in a blood chapter, how bilirubin, this greenish substance, is turned into a brown color, stercobilin, which gives our fecal material this typical brown color. Way back in a blood chapter, we mentioned vitamin K. This is needed for the proper production of those clotting factors. And in addition to dietary intake, our bacteria in our large intestine make this substance, and some vitamin Bs are also produced as a part of bacterial metabolism. In terms of movement of material from the lumen of the large intestine into the blood, we have about 10% of our water absorption. Electrolytes, sodium, potassium, especially absorbed fairly well in the large intestine, and some vitamins. On a previous slide we mentioned bacteria producing vitamin K, vitamin B types. Those vitamins are absorbed. The large intestine, and this is the clinical context here, large intestine is also used to provide medication for the body. So rectal suppositories are little jelly tablets or cone-shaped medication-filled containers, and they melt in the rectum, and their content can absorb, because in the large intestine, while very slow, we do have some absorptive activity. The fecal material itself is semi-solid, a paste-like material in the transverse colon, and when we are getting rid of this fecal material, we're getting rid of roughly about half of the material is bacteria, their products, undigested food components, and lots of epithelial cells from the large intestine. Here's yet another clinical context. These epithelial cells have their DNA, and if some of these epithelial cells turn cancerous, colon cancer can now be detected by analyzing DNA from fecal material. Cologuard is the name of the test. It's still fairly expensive. I'm not 100% if it's substituted for colonoscopy. I believe colonoscopy is still the gold standard, but there are people who are just scared of colonoscopy, or because of their health, it's not recommended. The epithelial cell DNA in the fecal material can provide at least some idea whether any of those cells are turning cancer. So these epithelial cells that we poop out can be used for analytical purposes. I also want to mention one thing here, that there is such a clinical procedure as fecal transplantation, where fecal material from tested and approved human donors are introduced into the large intestine. This procedure can re-establish of a bacterial balance in the large intestine. Clostridium difficile, C. diff is the other popular name for it. C. diff can overtake the large intestine and can kill the owner of that large intestine. C. diff spreads and establishes itself in the large intestine when treatment of the patient kills the rest of so-called good bacteria. So antibiotics, especially the more active ones, can upset the balance. So fecal transplantation may be an option for those patients. We have to talk about the defecation reflex. In the urinary system, we are talking about the micturition reflex, the reflex that allows us to relieve ourselves. Do a number one. Here we talk about what makes us think about doing number two. Well, our rectum has stretch receptors, and fecal material accumulating in the rectum will create nerve impulses. The nerve impulses are coordinated in the sacral aspect of our spinal cord, and from that control center, nerve impulses are sent to the diaphragm, abdominal muscles, and also to the opening of the internal anal sphincter. So the external anal sphincter is voluntarily controlled. The defecation can be postponed because we can override this defecation reflex, but when we are at a socially acceptable place, excuse me, this external anal sphincter, which is skeletal muscle, also relaxes and defecation is accomplished. Excuse me. So the last part of this lecture is about regulation of the digestive activities. So what's happening in our oral cavity, what's happening in our stomach, and what's happening in our small intestine can be regulated. Just overall comments for our stomach and intestinal activities. Stimulation always comes from cranial nerve 10, the vagus nerve. We'll see other influences as well. And in addition to the nervous system, surprisingly, we'll be seeing hormonal influences, both in the case of stomach being stimulated and coming from intestines acting on other digestive organs. So what's happening in our oral cavity? We have when we can see food, we can taste food or drinks, cranial nerve 7 and cranial nerve 9 will be sending stimulation to the salivary glands and saliva, and the amylase enzyme in there are going to be secreted into the oral cavity. Seeing, smelling, tasting food also activates the cranial nerve 10, the vagus nerve, and just by seeing, smelling, tasting food, our stomach churning, mixing, and gastric juice secretion will also be increased. When it comes to the stomach, positive influences, stimulation comes from the parasympathetic division of our nervous system. This is accomplished through cranial nerve 10. And as a consequence of that, we have mixing, secretion of gastric juice, and eventually emptying into the duodenum. Now, these are all activities that need to be stimulated through cranial nerve 10. We do have a hormonal influence portion here too. When we mentioned the stomach few slides ago, quite a few slides ago, we mentioned that some cells of the mucosa produce this enzyme gastrin. I'm sorry, this hormone gastrin. This is not an enzyme. So it produces the hormone gastrin, and gastrin further increases the gastric juice secretion and increases mechanical digestion in the stomach, churning and emptying. So both the nervous system, cranial nerve 10 mostly, and gastrin hormone is stimulating activity of the stomach. And here is the news about workshop number 3, the small intestine. So the small intestine is largely in protection mode. The small intestine can be overwhelmed by the acidic, corrosive chyme coming from the stomach. And so let's take a look at how the small intestine can put a stop to or slow down stomach emptying. Here's the first one, enterogastric reflex. This is based on totally nervous system and the local enteric nervous system neurons. So here is chyme filling the duodenum too much. Stretch receptors in our duodenum will be sending nerve impulses to the medulla oblongata, and from there sympathetic nerve impulses will slow down the movement of the stomach and also slow down the emptying, and this is accomplished by contracting the pyloric sphincter. So the enterogastric reflex starts in the duodenum and ends up slowing stomach movement and emptying. This is done in the name of duodenum protecting itself from overfilling and fearing that it would be overwhelmed by a large volume of acidic and corrosive kind from the stomach. A very similar kind of reaction is when hormones are used by the small intestine. So in addition to stretch receptors, the duodenum also has chemoreceptors. Too low pH will lead to this release of hormone secretin. Now secretin is trying to increase the neutralization of this acidic pH. So secretin is going to stimulate pancreas to produce more pancreatic juice, which will bring more of this neutralizing agent bicarbonate to the duodenum. And this hormone also puts a stop sign to the mucosa of the stomach, and so secretion of gastric juice, which has the hydrochloric acid in there, that will slow down. So through the hormone secretin, the small intestine is protecting itself from being overly acidic, and both the pancreas and the stomach are called in to help with this issue of too acidic chyme. And then here are chemoreceptors that detect partially digested or poorly digested proteins and triglyceride. And then the small intestine releases cholecystokinin hormone. This hormone will go to the stomach and slows emptying. Slowing the emptying of the stomach means that this material will spend more time in the stomach, hopefully increasing digestion. So we are going to bottle up this food and drink item in the stomach for more digestion. And then cholecystokinin calls on both the pancreas and the liver to from a pancreas to increase pancreatic juice production. This will bring in additional enzymes to deal with these partially digested food products, and increased flow of bile will bring in more of that emulsifier to again increase the digestion now in the small intestine of these partially digested triglycerides. The rest of this slide series are summaries. I want to click through this fairly quickly, but say a few words. So digestion of carbohydrates, you can see everything that's happening to carbohydrates here. I would add something, and I need to change this slide. Esophagus and stomach, this really should say that no new enzymes are added. Actually, salivary amylase is active in the esophagus as this bolus is pushed. In the stomach, there is no activity, so I need to adjust this slide. So in the esophagus, salivary amylase is still active for those few seconds that the bolus of food is pushed through. With the digestion of proteins, there's really not much to add here. Stomach, pepsin, and the set of protease enzymes from pancreas are responsible here, with a few last-minute degrading enzymes located in the brush border. Digestion of lipids. This also a slide that needs a little adjustment. The gastric lipase is missing from this list. So gastric lipase and lingual lipase will be degrading triglycerides to some degree in the stomach, although this is true that most lipid digestion happens in the small intestine. Pancreatic lipase is a very, very active triglyceride degrading enzyme. And this is, again, just for formality, for sake of completion, digestion of nucleic acid. We mentioned this a little bit when we talked about the pancreas. So I'm going to stop here, and this is the end of the recording on the digestive system.